So this is the next part of a series of lectures that we'll have, a series of modules, and this is called sequence analysis. Sequence analysis is essentially the key to bioinformatics because it was really the publication of the genome, the sequencing of the human genome, which really pushed bioinformatics as we've discussed before. It was the need for bioinformatics. It was the need for the use of information technology in the biological sciences, and on a grand scale, this is the publication of the genome, the work towards the publication of the genome when this really occurred. Um, Sequence analysis always has been the mainstay of bioinformatics. Very often, people will even ignore the proteomics aspects of bioinformatics. I remember the paper we looked at on the slide 2 modules ago about the paper by Mark Gierstein and his colleagues, where he kind of we had this where you had the genomics and the proteomics brought together as a confluence to create bioinformatics, right? And that is one of the definitions of bioinformatics that we accepted. So sequence analysis has always been the mainstay. What we're going to do in today's set of modules is look at sequence analysis as people originally imagined it, as it was practiced before. And when we think in terms of where bioinformatics is today, in terms of its scope, its size, you could sequence an entire genome in a day using high throughput next generation sequencing methods. What we're going to do today is a scaled down version of what is at the frontiers today. Sequence analysis, conceptually at least, is what you will see in next generation sequencing. Okay, so let's begin with sequence analysis. Now, one of the important aspects of sequence analysis is, you're going to from now on treat a DNA sequence or a protein sequence you're going to treat it as a string of letters. In DNA it's ATCG, RNA it's AUCG, and the 20 amino acids or the conventional amino acids in a protein sequence, and you're just going to take it for granted that, well, this is the sequence, which is what is called the primary structure. But I want you to begin to understand that while you write just a sequence of letters and say, well, this is my DNA sequence, there is a structure behind it. So at least, keep this at the back of your mind, that when you write A means something structurally, okay? So in order to understand that, let's look at the structure of DNA, okay? And so what you see are the different atoms that are represented in the structure of DNA, and these are color-coded. Not only are they color-coded, and this is typically convention. Oxygen is always red, nitrogen is blue, carbon is gray, hydrogen is a smaller version of carbon, phosphorus is yellow. And you can also see the relative sizes of these atoms. So what you see is a single helical strand, and DNA is a double helix. And so this is the double helix that you see. The way the helix is constructed, and it is sustained by what is called hydrogen bonds, which we will see in the next module, is a minor groove and a major groove. Similarly, a minor groove, there's a complementary major groove on the other side, and then there is a minor groove here. What we have is a sugar phosphate backbone, which composed of a five-membered ring. And we look at this structure in a little more detail. You'll see a 5-membered ring, ribose sugar, and there is associated with a phosphate group, so PO3 minus or PO4 minus. So this is associated with a phosphate group, and this forms the sugar. To the sugar phosphate backbone is attached a purine or a pyrimidine, and that is what constitutes the nucleotide. And all these nucleotides, if you consider the DNA to be, or even a single DNA strand to be a polymer, then the monomer is called. So when we write nucleotide, the primary structure of this compound, you will write it in the form of A, T, G, C, and some combination for for some length. So what while the sugar phosphate is common to the entire DNA strand, what is unique are these what constitutes the kind of the unique aspects of the nucleotide. So you have an adenine A, T is thymine, C is cytosine, and G is guanine. Now you notice that the pairing takes place. Again, these dotted lines they're not probably very visible now, but we look at it a little more clearly. This dotted line is a hydrogen bond. So an adenine will always pair with a thymine, and a guanine will always pair with a cytosine. And the next slide will show this. Next two slides will show this a little clear. So this is a purine. Adenine and guanine are purines, and thymine and cytosine are pyrimidines. Is a fused six-membered ring and a five-membered ring, and a pyrimidine is a single six-membered ring. And so the pairing is always exclusively AT or TA and CG or GC. A C will not pair with an A, and a G will not pair with a T. And remember, the pairing again is always purine pyrimidine. Let's make this a little less busy and just represent the sugar phosphate backbone with a ribbon, and then you will see that these are this is the purines and the pyrimidines. And so you have adenine, which is yellow, and guanine, which is red. And an adenine will always interact with a thymine, and this is kind of a conceptual representation that the fit is correct only between adenine and thymine, and guanine and cytosine. The other two do not fit. And these are what are called the base pairs. Okay, the base pairs, and the base pairs with its part of the sugar phosphate backbone is called a nucleotide. If we go a little deeper into it, you see the phosphate group is what links the ribose sugar PO4, and it's a ribose sugar is a five-membered ring which has an oxygen, and the rest of these are carbons. So you can look at this, and so we'll in the next picture we'll see what why sometimes you say, well, from the 5' end to the 3' end. The purine is a six-membered and a five-membered ring. It matches with a pyrimidine, which is a six-membered ring. Again, between the A and the T, you have two hydrogen bonds, and we'll see what that is. Between the guanine and the cytosine, it is also which is also a three hydrogen atom, which sustains this. Now remember, what sustains the helices, the double helices, the intertwining of this to create the double helical structure, is the hydrogen bond. There is no other way for these to actually interact with each other. Okay, now let's look at the sugar backbone. So in terms of conventional naming, you've got a five-membered ring, 1, 2, 3, 4, 5, which is an oxygen atom, and this is what is called a ribose sugar. And the naming is 1', 2', 3', 4', and 5'. And then from here, you will see the next ribose sugar. The next ribose sugar attaches here. And therefore, you will say this is 5', 3', so another 5', so the 5' end to the 3' end. In ribonucleic acid and deoxyribonucleic acid, the only difference between the two, there is one where the thymine is replaced by uracil. We'll look at that in the next slide. Is that deoxy, because the oxygen is removed from the 2' carbon atom. So 2' deoxyribose, that's the ribose sugar, and that's the DNA sugar phosphate. And in ribonucleic acid, there is an oxygen. So that is the two difference. That's the fundamental difference in terms of the backbone of DNA and RNA. Now let's look at the purines and the pyrimidines. So adenine and guanine are the purines. For example, adenine will only interact with thymine, or in the case of RNA, thymine is replaced by uracil, represented by AU. And again, six-membered ring, five-membered ring. What's the difference between adenine and guanine is that there is a carbonyl, oxygen is replaced by an amine group, and here, instead of a hydrogen, is replaced by an amine group. Those are the purines. In the pyrimidines, we have the difference between cytosine and thymine is an NH2 group. An amine group is replaced by a carbonyl oxygen, CO group, typical of ketones and aldehydes. and there is a methyl group here. Okay, so though that's the difference between thymine and cytosine. And what's the difference between thymine and uracil? Its replacement in RNA is that there is only one replacement, and that is the CH3 group in thymine is replaced by a CH group. So it's a smaller, because of the lack of an additional substituent, it's a smaller. So the structure of DNA or RNA, when you think about it, is that this hydrogen bond is what sustains the double helical nature of it. That's the reason why. So there is a certain kind of interaction which holds the two strands of DNA together or RNA together, and these are hydrogen bonds. So there's no real covalent bond. Now let's review a little bit about the differences between hydrogen bonds and covalent bonds and electrostatic bonds and coordinate covalent bonds. Okay, so just a quick review. A covalent bond is formed by the sharing of electrons between two atoms. Okay, and so that each of these completes the certain what is called a valency. And you guys can look this up in terms of what a covalent bond is. You don't see in organic kind of compounds, you don't see an electrostatic interaction where one atom actually donates another electron. So for example, sodium chloride, NaCl, one electron from sodium, which is electropositive, is donated to the chlorine. A bond is formed, it's pretty strong. Another type of covalent bond is called a coordinate covalent bond, and this is often seen in inorganic compounds. So where an organic compound is associated with a metal, it's called an organometallic compound. Again, I'm saying these things, you guys can do just a brief study, so you're able to separate an electrostatic, a covalent, a coordinate covalent bond. And in that coordinate covalent bond, there is a sharing of electrons, except rather than each electron being contributed by an atom towards a bond, you will actually see that both electrons are contributed by one atom, but they are shared like in a covalent bond, and therefore it's called a coordinate covalent bond. A hydrogen bond per se is not a real bond, but it is a mild form of an electrostatic interaction. Think of if you have the opposite poles of a magnet, this is what happens. So how is a hydrogen bond formed? So for example, here you will see that oxygen and carbon, the oxygen is what is called electronegative, and I'll explain this a little more. And when something is electronegative, it draws the electrons towards it, or the electron density towards it. So oxygen is a little more electronegative, which means that though we say that these bonds are shared, it draws this electron density towards this. So this becomes slightly more negative and this becomes slightly more positive. Similarly, you have this nitrogen of this amine group, this nitrogen in the amine group attached to a hydrogen, and nitrogen is a little more electropositive, and therefore what it does is it drags a little of that density away. It becomes slightly negative, and this slightly negative atom finds itself in the vicinity of a slightly positive atom, and there's that electrostatic kind of things that are not alike will interact. That's that interaction that you see between an adenine and a thymine. Two hydrogen bonds sustain it, and three hydrogen bonds sustain it. And when so hydrogen bond is essentially critical, literally critical for life. It's something that you don't normally think of because it's not a real bond, but it is very important. The fact that and we look at an example of water being sustained by hydrogen bonds is what allows it to be crucial to life.